A few videos ago, we were talking about the relationship of genes and the characteristics. As an example, over here, I mentioned before that, for example, a gene is a length of a DNA that codes for a polypeptide, and I told you that when the gene undergoes transcription, it produces the mRNA, and when the mRNA undergoes translation, it produces the polypeptide. And depending on the type of polypeptide you produce, it affects your characteristic. And I used this one example here where the polypeptides produce some pigments in the fur of this cat, and the cat becomes like a brownish color. The point I'm just trying to make here is there is a relationship where the gene will code for a protein and the protein affects the characteristic of the organism. So we are going to look at some specific genes that exist, and you also have to know these genes for your syllabus. And we are going to see how the genes affect our characteristic. And we are also going to talk about what happens when these genes undergo mutation. And we are going to see how mutation can also alter or change the characteristic of the organism. Now, the first gene that we are going to be talking about is something known as the TYR gene. So what exactly is the TYR gene all about? Now, the TYR gene exists in a lot of organisms, and it also exists in humans as well. So where exactly is the TYR gene located? As you can see, I'm showing you the list of the human chromosomes, all our 23 pairs of chromosomes. Now, I'm highlighting chromosome pair number 11 over there. And these pair number 11 over here is an autosome. It's not a sex chromosome, by the way. The TYR gene is located on chromosome number 11. You do not need to memorize that. You just need to know that the TYR gene is located on the autosome. It's not located on the sex chromosomes. So, what exactly does the TYR gene do? It is a gene that codes for an enzyme, and the name of the enzyme is called tyrosinase. Now, when the gene undergoes transcription and it produces the mRNA, and then the mRNA undergoes translation to produce the polypeptide. So I'm just going to put an arrow to just mention transcription and translation to just, summarize the whole process. So when transcription and translation happens, the polypeptide is produced where the protein in this case, or the polypeptide is called tyrosinase. So in this case, how does this protein tyrosinase affect the phenotype of the organism? So let's look at it. Now, you need to know what's the function of tyrosinase. Tyrosinase is an enzyme where it is able to, the active site of tyrosinase is able to react with an amino acid known as tyrosine. And when it reacts with tyrosine, it's able to produce something known as DOPA, D-O-P-A, all large letters, D-O-P-A. Interestingly, DOPA is also then able to react with tyrosinase, again, to produce something known as dopaquinone. The dopaquinone will then be converted into something known as melanin pigments. So, for the purpose of the exam, if a question asks you what's the function of tyrosinase, you have to mention that it converts tyrosine to dopa, converts dopa into dopaquinone, which is then converted into melanin pigments. If you forget the pathway, you might lose some marks, but you can also just say you might be able to get 1 mark if you just mention that tyrosinase is involved in the production of melanin pigments. But if I were you, I'll memorize the pathway where tyrosine is converted to dopa, dopa is converted to dopaquinone, dopaquinone becomes melanin pigments. So tyrosinase, the enzyme, is required for that first 2 chemical reactions. Now, how exactly does the melanin affect our phenotype? Very simple. It affects our phenotype by affecting the color of our skin, hair, and iris, where, depending on the type of melanin you produce as well, but to just make it simple, the higher the concentration of melanin you have in your skin, hair, and iris, the colors are going to be darker. So usually a person who has a light complexion on their skin would have less melanin. A person who has a darker complexion on their skin will have more melanin pigments. So now we are also going to talk about the situation where the TYR gene becomes mutated. In the earlier chapters in AS, chapter 6, I did mention that there were 3 types of gene mutation: addition, deletion, or substitution. You don't have to know that in detail. The point is, if the TYR gene becomes mutated, what could be the effect on the person's phenotype? Perhaps in some cases, if the mutation is significant, it is no longer able to code for tyrosinase. And again, because of mutation, it may change the mRNA codons. It may cause the primary structure of the protein to change, which will then change the tertiary structure of the protein. So look at the shape of the tyrosinase enzyme here. The tyrosinase, from having a proper active site here, the 3D structure of the enzyme has changed, so it becomes non-functional. What does it mean by it becomes non-functional? So look here. Can the enzyme react with tyrosine? Can the active site of tyrosinase react with the tyrosine amino acid? No, it's not able to do so. So would it be able to produce DOPA? No, it will not be able to produce the DOPA, and because there's no DOPA, it will not be able to convert it into dopaquinone, and because there's no dopaquinone, there will not be any melanin pigment production. So in this case, in the person who has a mutated TYR gene, what might happen is they will not be able to produce a functioning tyrosinase enzyme, and therefore it will impact their melanin production, where they will not produce melanin. And because they will not be able to produce melanin, what will happen then? The hair color might become pale, they might also have very pale skin, and they might also have very pinkish eyes, okay, pinkish iris. This condition over here, where the person has a lack of melanin inside their body, is known as something called as albinism, or a person who does not have melanin is referred to as someone who is an albino. So albinism is the condition where the TYR gene has been mutated, they are not able to produce the tyrosinase enzyme, or even if they produce the tyrosinase enzyme is non-functional. So melanin production stops, or there is no melanin production. So the phenotype of this person is they will have a condition known as albinism. The symptoms of a person with albinism is they usually have pale hair, pale skin, pinkish iris. They have a tendency to avoid very bright areas, and there are cases where people with albinism will have a higher chance of developing skin cancer if they are exposed to ultraviolet rays for a prolonged period of time. They have to be careful with that as well. And of course, it's also important to know that the normal TYR gene, which is not mutated, is dominant, which I'll refer to as capital A. The mutated TYR gene is usually recessive, which I'll refer to as small a. And because these genes are located on the autosomes, the males will have 2 alleles, the females will also have 2 alleles equally. And the genotypes, possible genotypes, can be large A, homozygous dominant, large A, heterozygous, or small a, homozygous recessive. If they have a genotype of large A, or large A, small a, the large A alleles are expressed, so the person's phenotype will have normal, so they'll have normal melanin production. But if they're homozygous recessive in this case, they will have a condition known as albinism. The next gene that we are going to be looking at is something known as the F8 gene. I did mention the F8 gene in a much earlier video when we were talking about sex-linked inheritance. The F8 gene is located on the X chromosome. The moment it's located on the X chromosome, you know that this is sex-linked, which means to say it will affect males and females in a different way. So, and I'm just drawing out the X chromosome over here. The F8 gene is that single line on the chromosome, and when the gene undergoes transcription and then the mRNA undergoes translation, the protein that is produced is called the factor 8 protein. Now, you don't need to go into the detail of this, but you just have to know that with the help of the factor 8 protein, the person's phenotype is they will have a normal blood clotting. Blood clotting just means when you have an injury, when you're bleeding, the bleeding must stop as soon as possible to reduce blood loss. So that's called blood clotting. Now, Because the F8 gene is located on the X chromosome, I also told you the F8 gene had two alleles in the previous video, capital H allele, which is normal, and the mutated F8 gene, we will represent it as a recessive allele, small h. And because it's located on the X chromosome, we write it as X capital H for the top one, X small h for the bottom one. Now, when you're writing the person's genotype, remember, males, if the person is male, the person will have one X chromosome and one Y chromosome. And if the person is female, the person will have two X chromosomes. We talked about this earlier in the previous videos as well. So the males can have a genotype of X capital HY or X small hy. You will not put the H alphabet on the Y chromosome because the Y chromosome does not have the F8 gene. So it's only located on the X chromosomes. But for the females, however, they can be X large H, X large H, X large H, X small h, or X small h, X small h. And in this case, the phenotype, the first male, they will have normal blood clotting. The second male, hemophilia. For the females, the first one is fine, no normal blood clotting. Second one, they will have normal blood clotting as well, but she's a carrier because she has one small h there. But the third female here, she will have hemophilia because she only has the recessive alleles. So this is another case as well that we have to know about how the F8 gene can affect the phenotype of the person. Okay, now what we are going to be looking at right now is we are going to be seeing the third gene, which is known as the HTT gene. Now the HTT gene is located on chromosome number 4, which I've highlighted over there. You don't need to know on which chromosome the gene is located. I'm just putting it as an extra bit of information here. Now the HTT gene is a very interesting gene. All right, the gene codes for a protein known as the Huntington protein. Now, here's the thing. We don't exactly know what's the function of the Huntington protein. That's the first interesting thing. But we think it affects brain development. Here's where it becomes interesting. All right. When you look at the HTT gene, I'm just drawing out here. The gene is quite long. I'm just drawing out part of the base sequences. The HTT gene has something called CAG repeats. And we don't exactly know why it has these CAG repeats. And these CAG repeats are called stutters. I'm highlighting the CAG repeats over there. And I want you to count how many CAG repeats are located. The amount of CAG repeats in this case over here, it has 21 CAG repeats. Now, 21 CAG repeats in the HTT gene is normal. It's harmless. However, in some cases, a mutation happens where addition occurs and the CAG repeats will increase, or the number of stutters will increase. In some HTT genes, they may have up to 36 to 39 CAG repeats, and in some cases, they can have up to more than 40 CAG repeats, as I'm representing here. Now, if a person, like I said, has 21 to about 35 repeats, they usually will have no problems. But if the person has 36 to 39 repeats, they may develop a disease known as Huntington's disease. And if they have more than 40 repeats in their gene, in the HTT gene, there's a very high chance of developing Huntington's disease. Now, what makes it even scarier is the fact that the disease will not show the symptoms when you are young. It usually shows the symptoms when you are older. In fact, in some cases, in a lot of cases, when the person has Huntington's disease, they will only start showing symptoms above the age of 30 to 40 years old. So the person, when they are born, they are fine, but as they become older, for some reason, the symptoms of Huntington disease starts to develop. The most common symptoms is it will start off with mood swings. Now, I know you might be thinking, I get mood swings. Does that mean I have Huntington's disease? No, of course not. But what happens is mood swings are the first symptoms, and then your body will start to have loss of muscle coordination, difficulties even holding an object, walking, or even sometimes talking, by the way. And of course, you will also get loss of cognition. Loss of cognition in this case just means your mental capabilities will start to fail. You will not be able to form coherent thoughts. You will not be able to, do calculations, even be able sometimes to talk to people or make decisions for yourself anymore. It will usually be fatal, and there's no cure for the Huntington's disease as of yet. Now, I don't mean to scare you even further, but in the first two diseases where we talked about albinism and hemophilia, those diseases were caused by the recessive allele. Huntington's disease is caused by the dominant allele, where if it's more than 40 repeats, we will represent that as capital H, and if the person has 21 repeats, that is actually the recessive allele. So here's the interesting thing. The normal allele is the recessive one in this case. The problematic allele is the dominant one. You see, so here, a lot of my students have this assumption that the good alleles are always dominant and the bad alleles are always recessive. No, they don't really care what is good or bad. It depends on which gets expressed over the other. That's the scary part. So, if the person is large H, they most probably will develop Huntington's disease. If large H, small h, again, even if heterozygous, the large H allele can express itself over the small h allele. So most probably develop Huntington's disease. But if they are homozygous recessive, small h, they will not develop Huntington's disease. That's the scary one right here. So, the next one that we also want to look at is, and we have actually talked about the HBB gene in chapter 6 when we were talking about sickle cell anemia. The HBB gene also, interestingly, is located on chromosome number 11. So it's located on the autosome. Just drawing it out over here, showing you the HBB gene. And the HBB gene codes for beta globin, which means to say when transcription and translation happens, beta globin will be produced. Now remember, beta globin is part of hemoglobin. Hemoglobin is a protein where it's made up of 4 polypeptide chains, 2 alpha globins and 2 beta globins. So the HBB gene is actually coding for just the beta globin. We don't have to care about the alpha globin in this case. Now, the beta globin becomes part of hemoglobin. It is assembled, the beta globin gets assembled into hemoglobin, and the hemoglobin affects the shape of red blood cells and oxygen transport. So if the person has a normal HBB gene, they will produce normal beta globin. So the shape of the red blood cell will be normal, biconcave shape, and oxygen transport will be fantastic. So I'm just drawing it out over here where the normal HBB gene, I will represent it as the HBA allele. That's the normal allele, where when it codes for beta globin, I'm just drawing out the polypeptide chain, everything seems normal over there. Now, in the mutated HBB gene, it was mentioned in chapter 6 AS that a base substitution happened. I did mention it then, and because of that base substitution in this HBS allele, one of the amino acid has changed, where glutamic acid in the normal protein has been substituted into valine as an amino acid, and I'm representing the valine as a red square. So only one amino acid has been changed in the entire beta globin. But by changing one amino acid in the protein, it causes a huge havoc in the shape of the red blood cell. So the shape of the red blood cell in this case becomes a sickle shape, and it will also transport less oxygen, and it may also cause blockage in the capillaries of the person, leading to blood clots. Because remember, the blood capillaries are very tiny. They are 7 micrometers, and at any one point, the red blood cells have to move in the capillaries in a single line. But because of this abnormal shape of the red blood cell, they may get stuck within the capillaries. They may clump together and form blood clot, and that's not good. Now, the interesting thing about the HBB gene and its mutated, the normal allele and the mutated form is, the two alleles are codominant. What does codominant mean? It means that both the alleles can be expressed if they are present. Okay, so the HBB gene is on the autosome, so whether the person is male or female, it it will affect the person equally. So if the person is HbA, the person is normal, okay, they will have normal red blood cells. And if the person is HbS, the mutated form, they will get sickle cell anemia. But because they are HbA, in this case, like I said, it's codominant. So during the case of codominance over here, what happens is both alleles are expressed. They will get very mild versions of sickle cell anemia. Very mild sickle cell anemia just means that most of their red blood cells will be normal, but they'll have a very low concentration of red blood cells that are sickle shaped. In most cases, they will be able to lead a normal life. In fact, in some cases, they might not even know that they have mild sickle cell anemia. So this is codominance of the HBB gene. And of course, the last gene that we have to talk about has nothing to do with humans. In fact, the gene here is in plants, by the way. And this gene is known as the LE gene. Now, the LE gene, which is found inside the plants, I'm just drawing out the chromosome here. The single line is the LE gene. The LE gene codes for an enzyme, which means to say when transcription translation happens, an enzyme is produced inside the plant cell, fine. You don't need to know the name of the enzyme. Don't worry about that. You do, however, need to know the function of the enzyme. What that enzyme is supposed to do is it's supposed to react with the substrate. You don't need to know the name of the substrate, but the product is important. It is able to form a product which is known as active gibberellins. And we have actually talked about gibberellins in chapter 15 and also chapter 15? Chapter 14? No, we talked about it in chapter 15, yeah. So we talked about active gibberellins in chapter 15. I'm going to put a link up there on the top right corner for your reference to see what's the effect of gibberellin. The LE gene codes for an enzyme, and the enzyme is needed to produce active gibberellin. That's it. That's what you have to say. All right. So why is active gibberellin required? Because active gibberellin, as I'm putting it over here, it stimulates the XET enzyme, another type of enzyme in the cell wall. It breaks the bonds between the hemicellulose in the cell wall. Cellulose microfibrils move further apart, which causes cell elongation, all right? And cell elongation will then cause the plant to become taller. That's what causes plants to grow. You see, for plants to grow, they need two types of enzymes, auxin and gibberellins, and gibberellins is equally important as well. So, the LE gene codes for the enzyme. The enzyme is supposed to produce active gibberellin, and the active gibberellin stimulates cell elongation. And how does it affect the plant phenotype? It makes the plant taller. Now, the thing that we have to talk about here is we have to then look at the mutation. So what happens if the LE gene undergoes a mutation? If it undergoes a mutation, it may code for a non-functional enzyme, which means to say if the enzyme is non-functional, I'm just going to show you like a distorted enzyme where the active site is completely, wonky in this case. So would it be able to react with the substrates? No. Would it be able to produce active gibberellin? No. Will it cause cell elongation? No. So what will happen to the plant phenotype? In this case, the plant will remain as a dwarf, or it will be short. That's it. So the LE gene in this case over here has two alleles, where large LE is a dominant allele where it codes for the normal enzyme, small LE codes for the non-functional enzyme. So if the genotype of the plant is large LE, will it be able to code for the normal enzyme? Yes. So will it be able to produce active gibberellin? Yes. Would it be able to cause cell elongation? Yes. So the plant in this case will be tall. Simple. But if the plant is small le, because remember it has two alleles here, it received one allele from the male parent, one from the female parent, and in this case over here, it cannot code for the normal enzyme, or the enzyme is non-functional in this case, the enzyme cannot produce active gibberellin, no cell elongation happens, the plant will be a dwarf. That's it. But if the plant is large LE, small le, it has one dominant allele, which I've underlined over there, large LE, and as long as it has the one dominant allele, enzyme can be coded for, normal enzyme is produced, active gibberellin is produced, cell elongation is produced, and the plant will be tall. So this is how the genes affect the phenotype of the organism. Oh, almost 30 minutes for this particular video, but the good news is we are nearing towards the end of this chapter already. I think I have maybe 3 more videos to do, and then we are done with this chapter. All right.